So today we're going to dive into a technique that's, believe it or not, both incredibly simple and seriously powerful. It's called paper chromatography. And trust me, you'll be amazed at what a little piece of paper and some liquid can show us about the world. I love this question because it gets right to the heart of it. You know that black ink you use every day? Well, it's usually not just one color. It's a secret mix of a bunch of different dyes. And with this simple paper trick, we can literally pull them apart and find the hidden rainbow inside. So, how in the world does this work? Well, the magic behind it all comes down to a really cool scientific idea. It's all about how different bits and pieces of a mixture react when they meet two totally different environments, or what scientists call phases. Okay, that's a mouthful, I know. Differential partitioning. But the idea behind it is actually super simple. Just picture you've got a mixture of things. They're going to separate because some parts are more attracted to a surface that doesn't move, while other parts would rather just go with the flow of a liquid that is moving. It's that difference in preference. That's the whole trick. And here's where it gets really clever. The stationary phase, the part that stays put, isn't actually the paper itself. It's the water molecules that are just naturally trapped inside the paper's tiny fibers. I mean, how cool is that? And the mobile phase? That's just a liquid like water or maybe alcohol that creeps up the paper and carries our ink along with it.
Yeah, think of it like a race. In your ink, you've got these different molecules. Some of them are just more social with that moving solvent. They dissolve really easily and are happy to get swept up for the ride. Those are the ones that race to the top of the paper. But other molecules, they're a bit more clingy. They have a stronger attraction to the water just sitting in the paper, so they get left behind. It's this difference in sociability that separates everything out into those nice, clean bands of color.
All right, so now that we've got the principle down, let's see just how easy it is to actually do this. It really is this straightforward. First, you draw a line with a pencil. You use pencil because the graphite won't dissolve and mess up your results. Then you put a little dot of your ink right on that line. After that, you just place the paper into a sealed jar with a little bit of solvent at the bottom. And this part's important. You have to make sure the ink spot itself is above the solvent. Otherwise, it'll just bleed into the puddle. Then you just seal it up and wait and let nature do the work. And check this out. This is the beautiful result. On the left, you can see the setup in the jar. And on the right, that finished piece of paper, that's called a chromatogram. You see the starting line where the ink spot was? Well, as the solvent traveled up, it pulled the mixture apart into components A, B, and C, and each one traveled a different distance. It's fantastic. Okay, this might just look like a neat classroom experiment, right? But the real-world uses for this simple trick are huge, and they're incredibly important. We're talking about an impact on everything from crime scenes to the food in your lunchbox. Seriously, this is used everywhere. Forensic scientists can use it to see if the ink from a suspicious note matches a pen found at a crime scene. Drug companies rely on it to make sure life-saving medicines are pure. Biologists even use it to separate out all the different pigments in a green leaf. And, as we're about to see, it's a huge deal for keeping our food safe. Let's really bring this home and talk about food safety, because this connects the science right to your life. You know the bright colors in your candy or in sodas? Well, food safety agencies use this exact technique to check for artificial dyes that are illegal or harmful. They can literally take the color out of a piece of candy and run a test just like this to see exactly what's in it. So what that means is this simple, cheap piece of science is actually a powerful guardian for all of us. It's one of the key ways we can be sure that the stuff we buy at the grocery store is safe to eat. In a way, it's a humble little piece of paper standing between you and some pretty nasty chemicals. It really makes you think, doesn't it? If a technique this simple can unlock the secrets of ink or protect our food supply, what other amazing truths are just waiting for us to find them, hiding in plain sight all around us? Thanks so much for joining me.